Hello friends, I hope you guys are doing well. So today we are in the third class of cytokinin series, and in the previous classes we have understood about the discovery of cytokinin and also about the biosynthesis of cytokinin. So today we are going to understand the role of cytokinin in agriculture and horticulture. So the first role, as we know, as the name suggests, cell division is the primary role of the hormone or the phytohormone. It is essential for the plant cell division. So it is written that cytokinin regulates cell division in shoot and root, because in the root tip and in the shoot tip the meristem is present, root apical meristem and shoot apical meristem. Meristem is such a zone where rapid cell division is going on. Rapid cell division is going on, and here the cytokinin is responsible for the rapid cell division. Okay, so it has been seen that in case of Arabidopsis, if we are increasing the level of cytokinin through genetic modification, the genes like KN81 and STM, which are responsible for meristematic function, is overexpressed. That means if we are increasing the cytokinin level, the genes through genetic modification, the genes responsible for rapid cell division in meristem, they are also being overexpressed. But in opposite way also, if we are overexpressing these genes KN80 and STM, these are also alleviating the cytokinin level. That means if these two things are mutually regulated, that means meristematic cell division as well as, or meristematic growth, as well as the cytokinin level, if one is increasing and another is definitely increasing, there is a mutual relationship or mutual reaction. Okay, and if conversely reducing endogenous cytokinin level by overexpression of cytokinin oxidase. Now this is important. Cytokinin oxidase is such an enzyme which is able to oxidize this cytokinin and deactivate the cytokinin basically. So these genes, if is expressed or overexpressed, reducing the cytokinin level, it will slow down the shoot development by limiting cell proliferation in shoot apical meristem. This highlights the direct role of endogenous cytokinin in cell division in apical meristematic zone. Apical meristematic zone. That means this paragraph is explaining that this KN80 gene and this STM genes, these two genes are responsible for your uh, meristematic growth, and this is regulated, mutually regulated by the cytokinin synthesizing gene. Okay, so and opposite is also happening. If cytokinin oxidase, which is reducing the endogenous cytokinin, it is also reducing the root and shoot apical meristematic growth. Okay, this is the first important role of cytokinin. Coming to the second important role of cytokinin, that it is regulating the cell division. How it is regulating the cell division? It is basically regulating the cell cycle. The reason behind regulating cell division is the regulation of cell cycle. So let's read this first. Cytokinin regulates cell division by influencing the cell division uh, cycle control. Okay, cell division cycle control. The the peak they pick the synchronized tobacco in S phase and the G1 phase. S phase is here in this picture. You can see the S phase is this synthesis phase, or otherwise here what is happening? There is DNA replication. Another is your G1 phase, where the growth and normal metabolic roles are going on. Okay, so we can say it as G1 because it is a first growth phase. So in two cases, in two stages, there is role of your cytokinin. That's why there is a peak. Both auxin and cytokinin actually regulate the cell cycle, but without cytokinin, auxin cannot process the cell cycle. Okay, so the cyclin dependent kinase. This is an important enzyme which is responsible, or enzyme group which is responsible for the cell division. These are regulated by auxin and cytokinin. Okay, so auxin, what is happening? Auxin is basically inducing the expression of CDC2, CDC2 mRNA, a major CDK, but the induced CDK remains inactive, alone cannot prompt cell division. Okay, so CDC2 mRNA is overexpressed or expressed through auxin, but the it remains inactive because of or in absence of cytokinin. But cytokinin can activate this CDC2 by phosphorylation dephosphorylation process. Okay, so cytokinin activates CDC25 like phosphatase, removing an inhibitory phosphate group from CDC2 kinase. Okay, CDC2 kinase, if the phosphate group is being removed from the protein, it is being activated, so potentially linking the cytokinin auxin cycle regulation. Additionally, cytokinins boost the overexpression of CDC, sorry, CYCD3, encoding a D-type cyclin crucial in regulating cell proliferation at G1 restriction point. G1 restriction point is somewhere here. Okay, in Arabidopsis, CYCD3 is expressed in proliferating tissue like shoot meristem and young leaf primordia. Overexpressing CYCD3 circumvent the cytokinin requirement for cell proliferation, indicating that one significant way of cytokinin stimulate cell division by enhancing CYCD3 function. Let's understand it in another way. Okay, so we understood two things. One is CDC2, another is CYCD3. Okay, CDC2 and CYCD3. Now we understood that CDC2 is activated, or it is processed by auxin, but it cannot be activated by auxin alone. It needs removal of phosphate group. So for that, cytokinin is needed. So CDC2 is regulated both by auxin and cytokinin, and another gene is there, CYCD3, which is completely regulated by your cytokinin and CDC2. Okay, and this is acting on the restriction location of G1. Okay, and what is happening basically, in broad sense, if we see, cytokinin is responsible for cytokinin oxidase synthesis, which is basically controlling the or restricting the cytokinin in the cell. But cytokinin, again, if it is there, it is controlling the IAA synthesis and its turnover. So this is basically a feedback mechanism, means cytokinin and auxin are controlling each other to regulate the cell division. Okay, this is the second role of or reason behind the cell division through auxin and cytokine. I hope this you understood very properly, because this explanation is not there in simple language in any book till now. Okay, so coming to the third role of auxin, that is cytokinin ratio and auxin, sorry, cytokinin and auxin ratio is basically regulating the morphogenesis in cultured tissue. If we see the thumbnail um, of this video, there is a test tube where the culture is there, and there one plant is there, or green tissues are there, and they are completely grown by cytokinin. There is very small amount of auxin. That's why there is no root development, only shoot development is there. Okay, so in this slide actually we are going to discuss that. Okay, the balance of auxin and cytokinin ratio in culture medium is significantly influencing the differentiation of cultured callus tissue from tobacco pith segment. High auxin and cytokinin ratio, auxin to cytokinin ratio is prompt for root formation, but if the ratio is low, it is going to lead the shoot formation. Okay, so that means auxin, if it is high, it is going to cause root formation, and if cytokinin is high, then it is going to go form shoot. Okay, and auxin, where it is synthesized, if we see the auxin synthesis, auxin is synthesized in meristems, apical root apical meristems and shoot apical meristems, but the major amount of auxin is synthesized in shoot apical meristem. So if you want to remember, auxin, if it is synthesized in shoot, it is enhancing the root growth. But in case of cytokinin, what is happening? The cytokinin is synthesized in root. That's why it is responsible for the shoot growth. Okay, so auxin is basically for root growth, and cytokinin is for shoot growth. Okay, so similar effects on morphogenesis based on auxin cytokinin ratios are observed in crown gall tumor resulting from mutants in the tDNA in Agrobacterium ti plasmid. Okay, if we observe the plant, some trees they have excess cell growth in the on the stem. Okay, so that excess growth of cell is basically the tumor, and that tumor is because of this agrobacterium, and the rapid cell division that area is because of this tDNA, or in the ti plasmid. So this ti plasmid is having two zones. One is TMR locus, another is your TMS locus. Okay, so from the name we can understand that this TMR locus is basically for root formation, and TMS locus is for basically shoot formation. Okay, or cell formation. Okay, so shoot cell formation. So in the TMR locus there is IPT gene. If we silent this gene, or if we produce mutant of this IPT gene, the TI plasmid inhibits the zeatin biosynthesis, causing the causing high auxin to cytokinin ratio, leading to root formation or root proliferation instead of undifferentiated callus. Okay, if we are going for, if we see the tumor, it is basically an undifferentiated growth, neither root nor shoot, only a mass of cells are there. Okay, so if in the TI plasmid, in the TMR locus, we are mutating the or silencing IPT gene, so what is happening, there is root proliferation in place of undifferentiated growth. But in case of such mutants where TMR locus is mutated, so auxin cytokinin ratio will be less, and there will be stimulation of shoot proliferation. Okay, these mutants result in partial differentiation of tumors known as tetratomas. Sorry, teratomas. We call it as a teratomas. That means there is partial differentiation in the tumor. Actually, tumor is undifferentiated, but because of this imbalance in auxin and cytokinin, there is differentiation. So we call it as a teratomas. Now, the fourth important role of cytokinin is, it is modifying the apical dominance and promoting lateral bud growth. What is happening in case of auxin? What was happening? There was apical dominance, so the apical part will grow ahead in one direction, but there will be no lateral growth. Okay, but in case of cytokinin, what is happening, it is controlling the apical growth, or inhibiting the apical growth and enhancing the lateral growth. It is happening in case of cytokinin. So let's read this. Apical dominance is significant factor shaping the plant form, refers to the inhibition of lateral bud growth by main central stem, while auxin primarily determines apical dominance. Cytokinin also influence lateral bud growth. Physiological studies demonstrate that applying cytokinins directly to axillary bud in various species stimulates cell division and encourage your lateral bud growth. Okay. Cytokinin overproducing mutants display phenotype supporting this observation. In wild type of tobacco, strong apical dominance limits lateral bud growth during vegetative development. However, in cytokinin overproducing plants, lateral buds grow vigorously, forming shoot that complete with the main shoot. As a result, these plants tend to have a bushier appearance due to enhanced growth of lateral buds. Okay, this is the fourth role of cytokinin, that it is controlling the apical dominance and enhancing the lateral bud growth. Coming to the fifth important role of cytokinin, it induces bud formation in moss. In lower organism also, it is responsible for bud formation. In moss, named as Funaria hygrometrica, it is responsible for the formation of bud. This transition begins with the swelling near the specific cell apical ends, followed by an asymmetric cell division, resulting the formation of initial cell that subsequently develop into bud, ultimately give rise to leafy gametophyte. Okay, here it is very important, in some cases the cytokinin is responsible for replacing light. So this is the example: light, particularly red light. Okay, is essential for bud formation in funaria. In the absence of light, bud doesn't develop. Okay. However, cytokinin, when added to the growth medium, can replace the need for light. Cytokinin not only stimulate normal bud development but also increases the overall number of bud formed. Even very low concentration of cytokinin at picomolar level can trigger the initial swelling at the apical end of specific protonemal cells, marking the beginning of bud formation. Okay, this is the fifth important role of cytokinin. Let's understand the sixth role, that is cytokinin overproduction has been implicated in genetic tumors. That means it has a practical role in genetic tumor formation. Okay, Nicotiana species can be crossbred to create interspecific hybrids. About 300 hybrids generated. Around 90% of the hybrids display typical characteristics of blending trait from both parent species. For instance, tobacco plants used for cigarette Nicotiana tabacum is such an interspecific hybrid. This should be in italics. Mm, I didn't change. This is a mistake. So, however, approximately 10% of these crosses produce offspring prone to spontaneous tumor formation. In these tumor prone hybrid, the level of certain compounds are notably elevated, around 5 to 6 times higher compared to the either parent species. That means tumor meaning what? Undifferentiated cell formation will be there. That means the compounds which are responsible for such formation, that means your auxin and cytokinin are high in these 10% of crosses. So this can be used for overproduction of your cytokinin in plants. Coming to the seventh important role, that is it delays leaf senescence. Cytokinin delays leaf senescence. This is very important. Once leaves are detached from the plant, they undergo a programmed aging. Okay, in animals we can call it as apoptosis, and in plants we call it as a programmed cell death. Okay, known as senescence, losing chlorophyll and RNA, lipid or other biomolecules which is being destroyed when aging process is going on. Okay. So cytokinin, when applied to isolated leaves, can significantly delay this senescence, especially when spread directly intact plants. Even a small spot treatment of cytokinin leaf remain green while surrounding tissue is and yellow. That means what is happening, if if a leaf is there, we put drops of cytokinin. So cytokinin drops will make the spots green, but the surrounding cells will become yellow. Okay, so this is happening in the cells. This kind of effect we call it as a Richmond-Lang effect. In the next slide I have mentioned it. Richmond-Lang effect. This is a very important question in exams. Okay. Mature leaves produce minimal cytokinin, relying on root-derived cytokinin to postpone senescence. In soybean, monocarpic senescence triggered by seed maturation can be delayed by seed removal, and seed pod control senescence by regulating root-derived cytokinin delivery to leaves. That means when one determinate plant is growing, the last part of the plant or last stage of the plant is the reproductive stage. So when seed formation is going on, there is yellowing and senescence. So to control the senescence, we can do one thing, that we can remove the seeds, or we can stop the seed formation by plucking it. So that way we can control the senescence and enhance the cytokinin level. Okay. Primary cytokinin involved in delaying senescence are gelatin riboside and dehydrogeatin riboside, potentially transported from root to leaves through the xylem via transpiration strain. Okay. To investigate cytokinin's role in leaf senescence regulation, tobacco plants were genetically modified to express the IPT gene under senescence-specific promoter. This lead to elevate cytokinin level as the leaves aged, effectively blocking senescence and limiting further IPT gene expression, preventing cytokinin overproduction. This experiment suggests that cytokinin naturally regulate the leaf senescence. Okay, so let's understand what is Richmond-Lang effect. This is very important for exam point of view. Okay, cytokinin wield a significant impact on delaying the aging process in intact leaves and various organs. Their role involves regulating protein synthesis and enhancing the transportation of nutrient through the phloem to aging region. So, what is happening when we are applying cytokinin in plants or in leaves as spots? The spots where cytokinin is there, it is controlling the protein synthesis. Also, it is transporting nutrient to that particular spot. That's how it is delaying the senescence and maintaining the greenness in that particular spot. Okay, the heightened nutrient accessibility leads to synthesis of new chloroplasts and a slowdown in degradation rate of chlorophyll within the leaves. Consequently, the delay of progress on the aging process. Initially observed by scientist Richmond-Lang in 1967, this phenomenon became known as Richmond-Lang effect. Richmond-Lang effect. Okay. The Richmond-Lang effect facilitated by cytokinin hold numerous practical applications. For instance, utilizing cytokinin can extend the shelf life of vegetable flowers by leveraging this delay in aging process. So this is one of the practical application of your Richmond-Lang effect. Okay. Okay, the next important role of cytokinin is it promotes movement of nutrient, as we read in the last slide. Okay, so when cytokinin is there, cytokinin is basically promoting the nutrient movement towards that particular area where cytokinin is high. Okay, cytokinin influence the movement of nutrient within plants, a process known as cytokinin induced nutrient mobilization. Experiment involving radio-labeled nutrients such as sugar, amino acids like biomolecules demonstrated that after treating a leaf or part of leaf with cytokinin labeled nutrient preferentially accumulated in the cytokinin treated tissue or parts. This hormone this hormone likely creates a new source sink relationship where cytokinin treated areas becomes the most attractive nutrient sink. Nutrient mobilization might occur because cytokinin stimulate the metabolism of the treated area encouraging nutrient to move towards it. Interestingly, even non-mobilizable substance, the non-mobilizable substance such as your sugar, are observed to accumulate in these cytokinin treated tissues, suggesting that metabolic process in the sink cell might not be necessary for this accumulation. Okay, this is how cytokinin is delaying the senescence and keeping that particular spot or that particular part young and active through attracting this nutrient movement by making it a preferable sink and other parts being the source. Okay, the ninth important point is the cytokinin is promoting the chloroplast development. This is also the same reason that when cytokinin is there, it is promoting the protein. As I told, protein degradation ko this is reducing the protein degradation and enhancing the protein formation. Okay, so this is how, and also it is attracting the nutrients. So chlorophyll synthesis will not stop in that particular area, making it green for a long time. So when seed germinate in darkness, resulting seedling appears to etiolate, displaying elongated hypocotyle and internode, undeveloped cytokinin, cotyledons and leaves, and immature protoplastids that form into etioplast rather than chloroplast. Etioplasts lack chlorophyll and essential components for the photosynthesis. However, when etiolated seedlings are exposed to light, etioplasts can mature into functional chloroplast. Treating etiolated leaves with cytokinin before exposure to light induces the formation of chloroplast with more developed grana. This treatment also enhances the synthesis of chlorophyll and photosynthetic enzymes upon illumination. Cytokinin, in conjunction with light, nutrition, and developmental cues, play a role in regulating synthesis of photosynthetic pigments and proteins. Exogenous cytokinin application mimic the effects observed when certain mutation lead to increased cytokinin level, aiding in de-etiolation of dark-grown seedling. This illustrates that how cytokinin contributing to promoting light-mediated development in plants. So, in the previous slides, we understood that cytokinin is, in case of mosses, it is replacing light. Not only in case of mosses, but also in case of plants, it is replacing light, because in plants, green plants, light is responsible for chloroplast synthesis. Okay, chloroplast synthesis, which is the reason of the greenness in the plant. Okay, so when one seedling is there, we are placing it in dark. What is happening? It will not be, the growth will not be proper. It will accumulate the succulency, and it will grow just in unidirectional direction, and there will be no branching, nothing. Okay, this kind of seedlings are called as etiolated seedling. Etiolated seedling, there is no chlorophyll, so the leaf will be yellow. So when such seedlings are placed in light, it will be de-etiolated. The process is called as de-etiolation. It will be de-etiolated and it will become green. Okay, the chloroplast, when it is placed in dark, it will become etioplast, but when it is placed in light, it is becoming chloroplast. But The theory is suggesting that this cytokinin can replace the light by application of this cytokinin to etiolated leaves before placing it in light. It will form chloroplast. Okay, so this whole theory is saying that cytokinin can contribute to promote light mediated development in case of plants. Okay, the 10th important role of cytokinin is it promotes cell expansion in leaves and cotyledon. Cytokinin notably promotes cell enlargement in cotyledon of dicot like mustard, cucumber, sunflower, especially during seedling growth. Treatment with cytokinin encourages additional cell expansion without increasing the dry weight. That means it will just accumulate um, the expansion of cell. It will not increase the dry weight, but it will just expand through accumulation of water. Okay, so in species when leafy cotyledon growth and expansion are more significant in light grown seedlings compared to those grown in darkness, cytokinin facilitate cotyledon's growth in both light and dark condition. Similar to auxin induced growth, cytokinin also induce expansion of radius cotyledon by enhancing the mechanical extensibility of cell wall. However, unlike auxin induced growth, cytokinin induced wall formation or wall loosening does not involve proton extrusion. This is very important. If anybody is asking ki cytokinin is also responsible for cell expansion and auxin is also responsible for cell expansion, what is the difference? So this is the basic difference, that auxin is involving the acid growth hypothesis by involving the proton extrusion, whereas in the case of cytokinin that is not happening. Interestingly, neither auxin nor gibberellin contribute significantly to cell expansion in cotyledon, highlighting cytokinin's distinct role in promoting cell enlargement in plant structure. The 11th important role is it regulates growth in stem and root. Endogenous cytokinins are crucial for normal cell proliferation in apical meristem, driving shoot growth. However, applied cytokinin often inhibit cell elongation in stem and root. For instance, exogenous cytokinin can hinder your hypocotyle elongation. What is hypocotyle elongation? It is the above ground part of your a below ground part of your seedling or the root part of your seedling, and it is the exogenous application is hindering this hypocotyle elongation even at concentrations that promote leaf and cotyledon expansion in dark grown seedling. Similarly, internode and root elongations are inhibited in transgenic plants overexpressing the IPT gene or cytokinin overproducing mutants. The inhibition of hypocotyle and internode elongation is due to excess cytokinin believed to link the ethylene production, showcasing the interconnectedness of hormonal regulatory pathways. However, studies suggest that at normal physiological concentration, endogenous cytokinin negatively regulate your root growth. Mutants with alternate cytokinin receptors or signaling elements, as well as transgenic plants engineered to lower cytokinin level, exhibit longer root compared to wild type. These findings indicate that potential negative regulatory role of endogenous cytokinin in root elongation. So before in the slides we understood that cytokinin is basically enhancing your shoot formation Also, cytokinin is reducing your root formation. This whole thing is about this. Okay, so this another important thing here is cytokinin, if it is overexpressed, it is leading to ethylene. Okay, this ethylene is basically, as we read here, that this ethylene is basically linked to your internode or hypocotyle elongation. Okay, or inhibition of your hypocotyle or internode elongation. Hypocotyle or internode and internode elongation means the root part of a seedling is being inhibited by excess cytokinin, which is leading to your ethylene production. Okay, so these are some of the in the 12th role we are understanding the some of the cytokinin regulated process in plants that overproduce cytokinin. The number one is plants with excess cytokinin basically display several distinct characteristics in increased leaf production from shoot apical meristem, leaves with elevated chlorophyll content appearing significantly greener, formation of adventitious shoot from unwounded leaf veins and petioles, delayed leaf senescence, reduced apical dominance. Extreme case shows stunted growth with significant shortened internodes and decreased rooting ability of the stem cutting, slower root growth rate. So these are the role of cytokinin which can be practically used by the agriculturist and horticulturist. Okay, so this is whole about the cytokinin and its role in agriculture. So if you were able to understand something, or the reasons behind the role of the cytokinin are understood, please share and subscribe the channel and like my videos and encourage me to do such work more and more. Thank you.